All right, now I'm very, very excited and honored to welcome Professor Akiko Iwasaki. Dr. Iwasaki is a Sterling Professor of Immunobiology and Molecular, Cellular, and Developmental Biology at Yale, and the Principal Investigator of the Howard Hughes Medical Institute. Professor Iwasaki received her PhD in Immunology from the University of Toronto and completed her postdoc training at NIH before joining Yale's faculty in 2000. Her research focuses on the mechanisms of immune defense against viruses at the mucosal surfaces and development of mucosal vaccine strategies. She's a co-lead investigator of the Yale COVID-19 recovery study, which aims to determine the change in the immune response of people with long COVID after vaccination. And she is the director of the new Yale Center for Infection and Immunity. Professor Iwasaki is at the forefront of COVID-19 pandemic with respect to research, science communication, and public service. She's quoted in numerous media outlets with expert insights and is considered one of the top 50 experts to trust during the pandemic. Professor Iwasaki is also well known for her advocacy on women and underrepresented minority in science and medical fields, and has a large follower base in social media. She has received many awards and honors. Just to name a few, she was elected to the National Academy of Sciences in 2018, the National Academy of Medicine in 2019, the American Academy of Microbiology in 2020, and to the American Academy of Arts and Sciences 2021. Professor Iwasaki's talk today is immunology of long COVID. If you can pull up your slides.
Yeah, thank you
so much for joining us. Please take it away when you're ready.
Thank you so much, Yating. That was a very kind introduction, and I'm very happy to be here. I'm going to share my screen. Okay, so you can see this, yes?
Yes.
Okay, great.
Thank you.
Wonderful. Yeah. So today I want to focus my talk on what we are understanding about the immunology of long COVID. Long COVID is a disease that occurs in about 10% of people who are infected with COVID, even with a mild infection. It is known to impact multiple organs, including the brain, lung, circulatory system, and the immune system. And my talk will really focus on what we can understand about their immune deviation that we can find in the long COVID patients. So before I get to long COVID, I also want to talk briefly about what we've discovered throughout the pandemic, particularly during the acute phase of the COVID. So we know that the SARS-CoV-2 infection can lead to very heterogeneous outcome, even within a particular demographic group. And so we can have anywhere between asymptomatic all the way to fatal COVID. And our work has demonstrated, for example, that immune signatures associated with severe and fatal COVID has to do with sort of misfiring of the immune responses, in that we found sort of a misdirected immune response indicative of sort of type 2 and type 17 immune responses that are normally not counterproductive during a viral infection. We've also seen that in the case of fatal COVID, that the kinetics of antibody responses, particularly neutralizing antibodies, were delayed. And these people, even though they developed the same amount of neutralizing antibody in the end, it took them over a long period of time to get to the level of neutralizing titers that are effective in controlling the virus and therefore succumbing to the infection. We've also demonstrated a lot of diverse autoantibodies that have functional consequence for the disease outcome of severe acute COVID, including those that target many, many immune factors, including interferons, cytokines, and cell surface markers. We've also been studying sex differences in the immune responses, particularly during the severe acute phase of the COVID, and have reported on significant changes or differences that we see between male and female patients. what we found is that in the female patients, we see an early activation of T cell phenotype that it corresponded with recovery compared to the male patients, especially as they age. We see a decline in activation of T cells that also correlated with the worse disease outcome. We've also been studying pregnant women who acquire COVID and have seen that in one case, a loss of the fetus during the COVID infection due to the invasion of placental cells, but particularly syncytial trophoblasts with the virus that resulted in severe inflammation in the placenta, leading to early termination of that pregnancy. We've also seen cases of pregnant women who are getting COVID during their pregnancy that ended up with a normal live birth, but however, we see significant interference signature within the placenta of these women, even though the virus replication is restricted to the respiratory tract. So there's definitely a distal impact of having SARS-CoV-2 infection on the distal organs such as placenta that could have some consequences later on. But today I'd like to focus on our ongoing work to try to understand how these long COVID disease manifests and try to understand the underlying biological mechanisms. So long COVID is not sort of unique in causing these post-acute infection syndrome, and that there have been many other pathogens, such as many viruses listed here, Ebola, dengue, polio, SARS, chikungunya, EBV, and many others, as well as non-viral pathogens, bacterial and parasitic pathogens that have been associated with these medically unexplained post-acute syndromes. And, you know, these diseases have, for the most part, been ignored and understudied, because it's very difficult to pinpoint the cause of these kinds of diseases, such as ME/CFS. However, the overwhelming literature is pointing to a potentially a common or at least overlapping causes of these diseases that occur with the trigger being very distinct. And this is a review that I was able to co-author with Jan Chotka, who himself has ME/CFS. And one thing that we are learning from this pandemic is the enormous resources that the patients themselves provide for understanding these types of unexplained diseases. And Jan is one of the people that I had the fortune of working with, but I'm currently working with hundreds of patients who have long COVID and other chronic issues, so that we can better understand these diseases. So the, clinical case definitions of long COVID differ between different sort of institutions. The WHO definition states that these types of lingering symptoms or long chronic symptoms starts within the first 3 months of the onset of the acute infection symptoms, and lasts at least 2 months, if not much longer, and cannot be explained by alternative diagnosis. Whereas the CDC version is slightly shorter, that people experiencing 4 weeks or more of these symptoms after the acute phase are considered to have long COVID. And then the NICE definition is a little bit longer, for more than 12 weeks being required. And I think the science is converging to sort of be more strict about these definitions, and likely 6 weeks or more, it sort of describes a better population of patients that really don't recover as quickly as those who have 4 or more weeks. But this is something that's currently being investigated. What is the best clinical case definition for long COVID, and how do we unify this? So a study published last year by Jim Heath's group really looked at what are the early factors that correlate with long COVID progression. And they studied COVID patients within the first 3 months from the time of acute onset, and then looked at a variety of factors within the first 2 to 3 months and sort of correlated with symptoms and factors that are identified from analysis of their plasma and blood. And what they found was that there are 4 post-acute syndrome of COVID anticipating risk factors associated during the initial diagnosis, which are type 2 diabetes, SARS-CoV-2 RNA-emia, Epstein-Barr virus viremia, and autoantibodies that are related to lupus antigens. So these 4 factors were identified during the acute phase to be predictive of those who develop long COVID. However, we need a lot more studies to understand, what are the factors that are driving chronic diseases at later phases of infection, and who recovers and who don't, and why. In order to provide not only insights, but therapeutic potentials for the people who have long COVID. So currently we're working with several hypotheses, 4 of which are listed here. I'm going to take you through these because I think it's important to consider these as a separate but potentially sort of synergizing driver of diseases. So the viral reservoir or viral antigens or RNA being sort of remaining in certain tissues could be one of the drivers of chronic inflammation. Of course, if there's a virus that's remaining that it cannot be cleared by the immune system, that will trigger chronic activation of lymphocytes as well as innate immune responses in response to, say, viral RNA. Second hypothesis is autoimmunity that's triggered by the virus infection. And these autoreactive lymphocytes can either have a sort of molecular mimicry between the viral antigens and the autoantigens, or it could be a completely unrelated specificity that's just triggered as a bystander. A third hypothesis is potential dysbiosis of the microbiome and or reactivation of latent viruses, such as the herpes virus family members. The fourth hypothesis is a unrepaired tissue damage, some inflammation that's triggered during the acute phase has a long-term impact on that tissue or even distal organs that can cause chronic inflammation and disease. And so these 4 hypotheses are supported by a number of studies. The viral reservoir hypothesis has over 100 different papers that's indicating the presence of RNA and protein or both, and in some cases, even infectious particles. The autoimmunity hypothesis, as I just mentioned, Jim Heath's Su et al. Paper initially identified autoantibodies, lupus-related autoantibody during the acute phase as one of the 4 predictive factors for developing long COVID. But at later points, I will show you some evidence today that at later points, we are not seeing autoantibodies as a major difference in long COVID versus control patients. There's also now at least 3 papers, if not more, indicating EBV reactivation, both during the acute phase as well as the sort of later phases of having reactivation of these viruses being much more elevated in the long COVID patients compared to the patients who control the disease and are recovering. And then there's also the tissue damage hypothesis that I mentioned. There are a number of studies obviously showing damage in various tissue from people who've had COVID months prior, but we also have an animal model which indicates that this could be possible to study. So I'd like to divide my data part of the talk into 2 separate parts. One, I will go over some of our mouse model of the mild COVID with prolonged central nervous system impact. And the second story is a yet unpublished study that looks at the multidimensional immune profiling of long COVID patients and identifying key factors that are distinct. So the first data study that I'd like to discuss is the one that was done in collaboration with Professor Michel Monge's group at Stanford, where we discovered a prolonged chronic changes in the CNS after a mild acute respiratory COVID. And this was made possible because we had an animal model to begin to dissect this. So how can a so-called respiratory virus impact the CNS? Well, one possibility is that there's a direct infection from the respiratory tract to the CNS, such as potentially through the nose, through the olfactory bulb into the CNS, and direct infection of the virus. And we have seen this in rare cases and in severe and lethal cases of COVID, but that's not what we focus on in this study. We also hypothesized that autoimmunity can target the CNS, which is also not a focus of this study. What we studied here is actually possibility of distal inflammation, such as that occurs in the lung, that may have an impact on the CNS. So this was made possible because we had earlier developed an animal model in which mice are transduced with AAV vector containing human ACE2 gene. And this, after about a couple of weeks, intratracheal delivery of AAV expressing human ACE2 enables the lung to express the entry receptor, human ACE2, for the SARS-CoV-2 virus. And we can inoculate these animals with SARS-CoV-2 virus intranasally, and then we can monitor the virus infection as well as immune responses. So because the human ACE2 is only expressed in the lung, there's absolutely no brain invasion of the virus, as you can see here. And there's also very limited sort of disease that we can measure. Body weight is essentially overlapping with the control, meaning that there's no weight loss, there's no sickness behavior as far as we can tell within the first 7 days of infection. Whereas there is definitely infection that's occurring in the lung itself. We've shown this earlier when we established the model, but you can see some of these viral antigens in the lung with this so-called mild acute COVID respiratory model. And so using this strategy, we confine the virus infection to the respiratory tract, but we're focusing on the brain, see what happens there. So that study, we first looked at both the serum and the cerebrospinal fluid from these animals at either 7 days post-infection or at 7 weeks post-infection on the right. And what you see is that during the 7-day post-infection time point, we see elevation of many cytokines and chemokines in the serum, as well as within the cerebrospinal fluid. You can see the red colors indicating elevation of these cytokines. But even at 7 weeks post-infection, we see elevated levels of these cytokines in this mild respiratory COVID model, both in the serum and in the CSF. And it's interesting to note that the CCL11, which is a chemokine that's associated with age-dependent neurological deficit, it is elevated in the CSF. When you look at the cross-section of these CNS tissues, what we found is the reactive microglia are abundant in the white matter of these animals. We used both 2 different types of mouse strains, CD1 and BOB-C, and looked at the white matter of these animals. And you can see that the mild COVID infected animals had elevated reactive microglia in both of these strains. And that was still observed at 7 weeks post-infection on the bottom here. You can see that in both the staining as well as enumeration of those staining. So clearly there's some sort of microglial activation that persists for 7 weeks post-infection. And in humans, autopsy studies of humans who died with SARS-CoV-2 PCR positive at the time of death, some people died of other causes, non-COVID related causes, without any COVID symptom at that point. However, just to take you through this, the gray matter has some limited increases in microglia, but it's really within the white matter of these patients that there's elevation of reactive microglia in these brain tissues. Again, indicating that even those who have no sort of overt COVID symptom at the time of death, they do have this elevation in white matter microglia reactivation. So what about other cell types within the CNS? We looked at hippocampal neurogenesis in animals that have had this mild respiratory COVID infection at 7 days post-infection on the top and 7 weeks post-infection on the bottom. And these purple sort of colors indicate the neuroblasts, which are dividing hippocampal neurons. And you can see that there's a significant reduction in these dividing neuroblasts in animals that had mild COVID, even at the 7-week time points. The other issue that I alluded to before is this elevated CCL11 in the cerebrospinal fluid of the mice was actually replicated in the patients with long COVID. This is a serum level of CCL11 that we measured from people with long COVID in collaboration with Mount Sinai Long COVID Clinic, which I will get into more detail in the second part of my talk. But here you can see that there is an elevation of CCL11 levels in those with self-reported brain fog compared to those without. The loss of oligodendrocyte is another feature that we see in the mild COVID animal model. So again, looking at the white matter oligodendrocytes at 7 days and 7 weeks post-infection, we see a significantly reduced number of these cells in the white matter, as well as you can see here in this section. And that consequently resulted in demyelination of the neuronal axons that you can see again here at 7 days, but also 7 weeks post-infection, likely as a result of the loss of the oligodendrocytes that are providing those myelin sheath to surround the axons of these neurons. So this study illustrated that even a mild and transient respiratory only SARS-CoV-2 infection can lead to prolonged changes within the central nervous system. And that really is, we think, is mediated by neuroinflammation. In fact, when we inoculated CCL11 intravenously into these animals, that was alone enough to cause the hippocampal neurogenesis defect. However, there are other chemokines, including CCL11, that also induce these reactive microglia phenotype that then we think leads to the dysfunction that we see, such as reduction in the oligodendrocyte precursors and oligodendrocytes, loss of axon myelination, and then reduced hippocampal neurogenesis, all leading to cognitive impairment. And this is important because Michelle Monge and I collaborated to begin with because Michelle treats or cares for cancer patients who develop these chemo brain, which is what happens after chemotherapy in certain patients. They have this neurocognitive deficit. And she saw a lot of similarity between the chemo brain patients and the long COVID patients, and wanted to understand if this is a possibility. And a lot of the insights that we are gaining here is based on her prior work looking at the chemo brain animal models. So it's really fascinating that we were able to, even though we're in a very different field, collaborate together to elucidate this potential pathway that's triggered during an acute respiratory COVID infection. So now I'm going to move to talking about our human long COVID studies. And this is, I'm just going to show you some key finding from our first study, but there will be many, many more studies coming out of these cohorts. So we had established several long COVID research cohorts. One is the Mount Sinai Yale cross-sectional long COVID cohort that I'm about to describe to you today. We have also established Yale recovery cohort that Xueting was referring to in the beginning of her introduction, where we are studying people who have long COVID who had then gotten a vaccine and seeing how their immune response and symptom changes. The third one is called the Yale Listen study. This is a symptom-based long COVID cohort, which also includes post-vaccine syndromes that some people are suffering from. There seems to be a lot of similarities between long COVID and post-vaccine long haul. Then there's this long impact that we have started at the very beginning of the pandemic. We developed a impact Yale study cohort that we are following now over 3 years and trying to understand what the acute phase changes in the immune system might impact their recovery and long-term chronic issues that people develop. And so these are the 4 of the cohorts that we are starting now. But today I want to really focus on the first one, which we have made, I think, the most progress so far. And this is the Mount Sinai Yale Long COVID study, in which it's a really an equal partner, Dr. David Petrino, who's a director of the rehab center at Mount Sinai, who cares for thousands of long COVID patients and has been an advocate of long COVID research since the beginning of the pandemic. His team, Jamie Wood, Laura Tabakoff, and Dana McCarthy, collaborated with us at Yale to really deep dive into the immunology of long COVID. And John Klein is the lead first author of the study, but was assisted by Rahul, Peiwen, Jillian, Jeff, and Sasha as co-first authors of the study. And David Van Dyke is a computational scientist that we're collaborating to do machine learning with. And Aaron Ring developed this rapid cellular antigen profiling, which enabled us to do in a high throughput manner, look at autoantibody reactivity in these patients. So this my long COVID study design is to do a cross-sectional study of EMR and symptom survey, as well as a variety of sort of physiological analyses, including plasma proteomics, phage display library to look at antibody reactivity to a variety of antigens, the human exoproteome through the REAP analysis that Dr. Ring's lab developed, flow cytometry to look at activation and cell composition in the periphery, as well as looking at SARS-CoV-2 antibody. We had 99 long COVID patients, about 40 convalescent controls. These are the people who acquired COVID around the same time as the long haulers, but have fully recovered. The healthy control are the ones who've never been infected with SARS-CoV-2. And the healthcare workers, these are the Yale healthcare worker cohort that we've been studying their immune response against vaccines and antigens. So in terms of the demographics, the typical age of these people are, the median age was 45.8 for the long COVID and 41.4 for the convalescent control. The sort of high risk age group for long COVID that develop after a non-hospitalized infection tend to be around 36 to 50 years of age. But of course, it spans age groups below and above. The sex of these patients are predominantly female. This is very typical of other long COVID studies as well. Long COVID in that group is really predominantly female. Acute COVID severity, most people were not hospitalized. We focused on these people because the severe COVID leading to long-lasting impact probably has a different physiological mechanism compared to mild COVID that turns into long COVID. And again, there was no statistical difference between the convalescent control and long COVID for these parameters. And days from symptom onset was about 400 days plus or minus in both cases. So this is really looking at, this is one of the sort of the latest time point study that's published so far. Ours isn't published yet, but that's been sort of reported so far. So we're looking at much later time point than the other studies of long COVID. So the long COVID patients have significant perturbations by flow cytometry. And we have looked at, hundreds of different cell types, but I'm only reporting the ones that are significantly different between the groups. So the non-conventional monocytes are elevated in long COVID patients. And then the conventional dendritic cell type 1, these are the dendritic cells that are key in priming cytotoxic T cells and Th1 cells. That seems to, the circulating levels is much lower in the long COVID compared to the convalescent control in yellow and healthy control in orange. Meanwhile, activated B cells and double negative B cells were elevated in long COVID patients. Long COVID patients also have changes in their T cell compartment. So while the TEM numbers are similar, the central memory circulating CD4 T cells are lower in the long COVID, whereas the exhausted T cells that are either CD4 or CD8, they're both elevated in long COVID patients. And this to us indicate that there must be some chronic antigen that's continuing to stimulate these T cells, and that is not being able to be removed from the body. Now, we don't know what the nature of that antigen is yet. We are doing tetramer staining to determine the specificity of these T cells. It could be SARS-CoV-2, it could be autoantigen, it could be reactivated EBV or other things. We don't know yet. The long COVID patients also have elevated type 2 T cells. So here we did intracellular staining after an in vitro stimulation of T cells. And what we're seeing is elevation in interleukin 2, 4, and 6, as well as some polyfunctional T cells that secrete both IL-4 and IL-6, both in the CD4 and CD8 compartments are elevated. In fact, these cells are pretty much only found in long COVID patients. So this is unusual for a sort of a viral infection because we expect more of type 1 immune responses to be elevated, but instead we are seeing these type 2 immune responses being elevated. Along with the T exhaustion phenotype, there seems to be some sort of immune deviation that's occurring in these patients. We also looked at antibody responses against SARS-CoV-2 antigens, and what we are seeing is that there is a significantly elevated anti-spike proteins, S1 and receptor binding domain, in long COVID patients compared to the convalescent control or the healthy control. And these participants all received 2 doses of mRNA vaccines around the same time. So it's not the vaccine differences, but there's something intrinsically different about the long COVID patients that have this elevated levels of anti-spike antibody. But when you measure the neutralization capacity, they're poorly neutralizing. So this sort of suggests that there may be chronic antigen that's stimulating the elevated levels of these antibodies over time. What about other features that can be measured from the plasma? So we looked for cytokines and hormones that are distinct in long COVID patients. And the top finding we have is the difference in the cortisol level. So this is looking at negative log 10 p-value with ANOVA. And essentially, cortisol is the most significantly different factor we find. And in fact, the long COVID patients have about half of the level of cortisol as the healthy control you can see here. So cortisol is an important hormone that's regulated by the hypothalamus pituitary adrenal axis. the fact that we see lower levels of cortisol, we thought may induce compensatory increase in the ACTH level to try to restore that level from the adrenal glands. However, when we measure ACTH level, the long COVID patients have similarly low levels of ACTH compared to the other groups. This suggests that the defect might be actually upstream of the adrenal glands, in that there may be some sort of pituitary or even hypothalamus defect that's leading to this low levels of cortisol. The important thing is that because cortisol is a diurnal hormone, we need to make sure that the collection time is about the same in different groups, and indeed they are about the same. There's no significant difference in the collection time. And long COVID status is the most distinguishing factor for long COVID that we can measure from the plasma. However, we also see elevation in the level of IL-8 and other chemokines and even C4b, a complement factor. So there may be other things that are going on that's indicative of inflammation. But again, it's not the sort of typical type 1 interferons and interferon gamma that we see during an acute viral infection. What about reactivation of latent viruses? We did not study the microbiome in our study, but we are looking at reactivation of viruses. And using this REAP strategy, the Ring and his lab member, Jill Jaycox, found that there is elevation of antibody against certain EBV antigens, as well as the varicella zoster virus antigen GE in the people with long COVID. There was also an HSV antigen that was reduced in long COVID patients, but when we compared these levels in seropositive patients alone, that difference went away, meaning that the real differences are restricted to the EBV and VZV antigens. If you look at the latent infection status of these patients, there is no significant difference between control and long COVID groups. So they're like, typically there's over 90% of adults are infected with EBV, and there's really no difference in the seroprevalence of these EBV in the long COVID patients. So what this is indicating to us is that there may have been a recent reactivation with EBV that led to the elevation of antibody against these lytic proteins. And we actually confirmed this using two other orthogonal methods, ELISA as well as a Serimmune, which is a linear epitope mapping strategy on bacteriophage. And that also resulted in elevation of antibody against a linear epitope within the GP42 of EBV, as well as VZV-GE, as I mentioned, is elevated in long COVID patients. And interestingly, the double positive CD4 T cells that I mentioned earlier, the IL-4, IL-6 double positive, that level, it actually correlates with the amount of IgG against EBV glycoprotein P23 or GP42, suggesting that the type 2 T cells that we see may be reflective of the reactivation of EBV. And it's well known that this, especially the GP42, is a viral protein that's used for entry into B cells. And that molecule actually interacts with MHC class 2 to reduce the Th1 immune responses in the interacting CD4 T cells. So this may be reflective of this kind of interaction between EBV reactivation and T cells that are responding to it. But again, we need to do tetramer staining to make sure that that conclusion is actually valid. So what about autoimmunity? So we have, again, used this REAP strategy, which found a number of autoantibody, as I mentioned earlier, during the severe COVID phase on the long COVID patients. And we do not see a significant differences in the number of autoreactive antibodies per patient or the number of reactivity against the long COVID propensity score. This is based on patient-reported outcome severity. And you can see that there is no correlation between these parameters in long COVID patients. And this is looking at a variety, panel A is looking at autoantibody against a variety of antigens that are listed on the left in the convalescent control, healthy control, or the different clusters of long COVID patients. And again, we don't see a significant differences between these reactivities. So using the variety of different biological parameters that we measured, we used a machine learning strategy to look at whether these immune phenotypes alone can classify long COVID. And essentially, the machine learning was able to classify the long COVID patients based on immunological parameters alone with the 94% accuracy. And the lasso regression that was used to identify factors that led to the classification really highlighted not the autoantibodies, but things like the anti-SARS-CoV-2 spike antibody, as well as EBV reactive antibodies, circulating cell differences, as well as factors in cytokines and cortisol. And when you look at this a map of what's elevated and what's reduced in long COVID, you can see that the cortisol is really the one that stands out as being quite different in long COVID patients, that's being lower in the long COVID patients. And there are other factors such as the effector circulating central memory T cells, CDC1 levels, and CD8 T cells, for example, are slightly lower in long COVID, significantly lower in long COVID. Whereas what's higher in long COVID are things like the EBV reactive antibodies, CD4 T cells, and effector type 1 CD8 T cells, and galectin-1 and other factors. So this is sort of showing us like what's the most significant changes that can result in classification of long COVID from long COVID. And we also have begun to use the same set of data to look for sex differences in long COVID. And this is led by Julio Silva and Takahiro Takahashi. It's not even on medRxiv yet, but just to give you a sense for some differences that we're seeing in male versus female patients. What we're seeing is that there are distinct symptoms that are associated with sexes. So the graph here is indicating the blue indicates female and pink is male. And you can see that more frequent in female is many different symptoms that are distinctly more frequently in female, such as dizziness and body temperature issues, cough, throat and chest pain, and so on. The only thing that was elevated in the male predominant symptom was sexual dysfunction, whereas hair loss was the most significantly sex different symptoms that was reported predominantly in female. So these symptom comparison alone is telling us there's definitely a female dominant symptoms that are associated with long COVID, and that we need to study their immune responses also in a disaggregated manner. Again, looking at symptom burden, total symptom burden, the female patients have elevated level compared to male, and organ system involvement, again, females have more than male. So this is sort of beginning to show us some differences that are associated with the symptoms as well as symptom burden. And now we need to understand the immune responses that deviate between these sexes. So putting all this together and coming back to my original hypotheses, what we're seeing is that the viral reservoir hypothesis is still definitely a possibility based on the exhausted T cell phenotype, as well as the elevated antibody levels to the spike protein, as well as many others have demonstrated, as I mentioned, chronic antigen and RNA in various tissues from people who had COVID months prior, as well as David Wald's group showing the elevated circulating levels of spike protein in people who have long COVID. All of these kind of suggest the presence of viral antigen and potentially RNA. So we're going to start a Paxlovid clinical trial through the Yale Lesson study that I mentioned. And this is a key collaboration with Harlan Krumholtz and his team. And we are hoping to start the recruitment as early as next month. And with respect to autoimmunity, at least autoantibodies to human exoproteome is not a significant factor that distinguishes long COVID patients in our study. So it's not likely that long COVID is predominantly driven by autoantibodies. However, we haven't ruled out intracellular antigens and also T cells reactive to tissue antigens. So it's still a possible sort of cause for long COVID. We have to investigate more. With respect to latent virus reactivation, this is also becoming a viable sort of thing to look into because of multiple reports, including ours, that show reactivation of EBV. And because EBV is also associated with a number of autoimmune diseases, this in a long term, it's possible that these reactivation could lead to autoimmune sequelae, but that will likely take years and years to develop. Anyway, this is something that we need to understand, whether it's a chicken and egg question. Did the latent virus reactivation happen because of long COVID, or is the reactivation leading to long COVID? These are things that we don't quite understand yet. And then tissue damage, as I mentioned earlier with Michelle's group, we are now starting to study how do we prevent this sort of brain fog issues that are what we're seeing in the animals with vaccinations or treatment or drugs that target the reactive microglia. But obviously we need a lot more sort of in vivo human analyses in order to understand how these tissue damage might be leading to long-term consequences. So what we need now is a deeper understanding of pathophysiology of long COVID. So the viral reservoir driving variety of different sort of gears that are listed here. It could be sort of tissue damage that's leading to latent virus reactivation, so on and so forth. We don't quite understand the direction of the gear or if there's any kind of connection between these gears, but these are something that we need to study. And downstream of all these root causes, we are seeing quite common things like vascular damage, microclots, platelet activation, hormonal imbalance, and potentially mitochondrial dysfunction. And what downstream of that are all these different symptoms that we are seeing with post-exertional malaise and dysautonomia, POTS, many other symptoms that are being reported. And then ultimately what we need is like sort of biomarkers to identify these different endotypes of disease caused by different root causes so that we can start to treat patients that target the right kinds of pathological agent. Randomized clinical trials and prevention is really important as we speak. And I think randomized clinical trial with placebo-controlled together with biomarker analysis to inform what are the biomarkers of responsiveness and how do we future classify patients who might benefit from one drug versus another. These are all very important questions that we need to address going forward. And I just want to end here by thanking the numerous individuals who contributed to this study that I was able to share with you today. Lab members that I highlighted already are bolded here, but there are many others that contributed for collecting the samples and archiving and biorepository creation, as well as the Yale Impact team, who really contributed this repository that's become so useful for the investigators to understand acute and now long COVID diseases. the David Petrino and his team, just absolutely inspiring to work with this team from the Mount Sinai. We also have the Yale Lesson and Recovery cohort, again, led by Harlan Krumholtz and many others who contributed. We are continue to do immune profiling and data analysis using the platforms that we developed and are collecting more. Aaron Ring, Ruslan Mejtov, David Van Dyke, Wade Schultz, and the Ceramine team have been instrumental. We are expanding our analysis to ME/CFS and other post-acute infection syndrome, and then looking at the CNS inflammation and potential disease altering drugs with Michelle Monchi's group. So I'm going to end here and take any questions.
Thank you so much, Professor Iwasaki. It's so wonderful talk. Now we open the floor for questions. You can either send it through the chat and let me know whether you want to ask your question by yourself, or you can type in, I will read for you. To start with, I have, this is so fascinating. I always found immunology and infectious disease are just fascinating work. And also, not to say like it's related to the long COVID challenge we had during the pandemic. So my question is that it's also the gender difference between the immune, there's surely some immune response difference between the genders. But I was wondering, like in the cortisol level in the previous long COVID cohort, did you find there's a difference between genders in the long cohort patients?
Yes.
Level.
Yeah, definitely. So if you look at studies across the world, female sex is definitely predominant. And as well as this age group that I mentioned, 36 to 50, that is a kind of a dominant risk factor age group. So that's sort of what we're seeing in the clinic, as well as we try to sort of reflect the community in our recruitment. But yeah, that's definitely, there's a sex difference, sex bias in long COVID.
Great. Any questions from the audience? Feel free to raise your hand or form your question in the chat. Yes, Bill, please.
Hello, thank you very much. I was typing my question, then I sort of thought, well, I could just ask it. That was a fascinating presentation. I've got actually two questions. The first one is, I'd love to know what you think about the potential for there being more than one sort of etiology within the class of long COVID following mild infections. Because, you know, obviously, as you said, severe infections have, people have problems as a result of ventilation, which might make it challenging to identify some of the specific immunopathology. And the other thing is long COVID following reinfections. I mean, if there's any predisposition, we do expect the frequency of long COVID to decline over time as people tend to develop it. So anyway.
That's right. Yeah. So thank you so much, Bill, for these questions. Yes, I believe there is more than one etiology driving long COVID that happen in a non-hospitalized COVID patients. That's why, we're trying not to kind of narrow down too early on one hypothesis versus another. But the persistent virus antigen is definitely seen. Whether that's driving the disease itself is not known, right? So is it sufficient to cause disease? No. Is it required? We don't know. So that, there's a lot of questions, but it is seen. So that is why we're doing a Paxlovid trial, but we also want to do, many other trials based on the ideas that we're developing, right? So, but this is the one that we can do, I think, the earliest. But definitely more than one etiology. And in fact, it may not even happen simultaneously, right? So maybe some sort of inflammation that drove reactivation of latent virus, then that contributed to autoimmune disease. I mean, there are lots of directions that one can take in a subsequent sort of sequential manner. So that's also something that we need to pay attention to. What time point are we looking at? And, because 8 months is different from 12 months is different from 24 months. So it's a long answer to your question, but that's how we are thinking about it.
I'm really glad to hear about the Paxlovid trial, because I remember saying to somebody when you first heard about those little bits of spike, saying somebody should try Paxlovid, and people just said, well, yeah, we only have anecdotal reports that people seem to say they've felt a bit better after taking it when they've been reinfected. But yeah, as I said, do you have any sense of long COVID and reinfections?
Yeah, that's a very kind of heated topic in the field.
Yes, you might, that's one way of putting it.
Thank you. So, okay, if you are a person who, let's say, recovered without any symptom from the first infection, reinfection will not necessarily increase, I think, the chance of you developing long COVID. However, it's not universal. There are people who recovered with mild COVID, no problem, the first time around, who got second infection and third infection, then got long COVID. That is a possible path, but maybe less likely than people who get even a milder infection the second time around. However, if you developed long COVID with the first infection, your likelihood of making that worse with the second infection is probably higher. So if you've already developed long COVID, having second and third infection is likely going to make things worse. Whereas if you recovered fully from the first infection, the second and third infection may not be, as high risk for developing long COVID. I mean, this is all sort of, again, I don't have a real evidence for any of this, but this is what I'm seeing.
Absolutely. And it would make sense with some of the things that you were talking about in terms of some of the markers, the biomarkers that you found. And I guess just thinking now as we're talking, it's possible that somebody could have COVID, recover, make a full recovery, and then get EBV, and then later on get COVID, and anything which was mediated by EBV could then be following. Anyway, fascinating. Thank you so much for coming and talking to us about it.
Yeah, thank you so much. Exactly. I'm very interested in this sequential outcome of sequential infections, but there really isn't great evidence out there for these things. Yeah.
Thank you so much for the discussion. For the sake of time, I think we're gonna end here. And I just want to forward some of our participants' message saying beautiful work and a great presentation. We all agree on that. So to close out is that on behalf of the Center for Communicable Disease Dynamics and the Harvard T. Chen School of Public Health, let's thank Dr. Iwasaki again for joining us today and for this fantastic talk. Thank you so much, Dr. Iwasaki. And for the audience, please do get in touch with any of the seminar organizers if you have questions for us or for our speaker. And also please check out the CCDD website to stay up to date on upcoming seminars and other events, and also feel to check out the recorded session for this talk and for previous seminars. We will not have the seminar next week because of spring break. We will resume the week after. Thank you so much for joining, or we'll stay in touch in the future. Bye.
Thank you.
Thank you.